Okay. So today we will discuss about the culture media. Okay. So what is culture media actually? So they are some ordinary medium. Okay, used to grow the bacterium from the infected material or from the clinical samples. Okay. Last time we discussed about the culture method. Okay. So culture method and culture media. Okay. Both are linked together. Okay. So culture media, uh, maybe they can liquid, they can solid, they can semi-solid, and some anaerobic bacteria is there, aerobic bacteria is there, special media is there. Okay, and enrichment, special, indicator. Okay, so many culture media is there. Okay, so we will discuss today. We'll discuss about the type of culture media basically, and what is the uses of culture media, their compositions. Okay, why we are saying this is indicator media, this is enrichment media, then this is enriched media. Okay, and why this is selective media, what is the importance of selective medias. Okay, so we will discuss one by one. Okay, so let's start. So basically, uh, I already told you about the definition. Culture media are required to grow the organism from infected material to identify the causative agent. Causative agent means microorganism that can cause the infection to the human beings. Okay, so so many bacteria, viruses, fungus, and parasite is there. Okay, that's come under the microbiology, and they affect the human beings. Okay, so what is there is basic constituent. Okay, so where we start to prepare the culture medium. So some basic constituent is there. Okay, so a culture media required. Okay, like water, source of hydrogen. We can say distilled water or potable water with the low mineral. Okay, we can only remember about the water electrolytes like, okay, like NaCl or other electrolytes. Okay, and peptone. Okay, so peptone is the complex mixture of partially digested protein. So basically basic components means हमको किसी भी मीडिया को बनाने के लिए इसका जरूरत पड़ेगा ही पड़ेगा। ठीक है, इसी से हम स्टार्ट करेंगे बनाना। ठीक है, कोई भी मीडिया है हमने बनाना है तो हमको ये सब चीजें चाहिए ही चाहिए। And peptone, what is the source of peptone? Obtained from lean meat or the protein materials like casein and fibrin. Okay, some constituent like it contains proteases, amino acids, inorganic salts like phosphatase, potassium, magnesium, and growth factor like nicotinic acid and riboflavin. Okay, some bacteria need some special vitamins also to their growth. Okay, and we use blood or serum. Okay, in culture media we also use, this is also a major components in the culture media. Okay, like blood or serum. Okay, so we use 5 to 10% sieve blood. Okay, in the case of blood we use 5 to 10% sieve blood. Okay, now there is agar. Okay, so what is agar? Basically, if we talk about the agar, so we can say in a single word, it is a solidifying agent. Okay, solidifying agent of a culture medium. So agar is a solidifying agent. We can saw the first statement, available in powder form, prepared from the cell wall of seaweeds, red algae species, Gelidium and Gracilaria. So this is about agar. What is the components? It contain mainly cell wall derived long chain polysaccharides like D-galactopyranose and concentration of agar. So this is the important. What is the composition of agar? So if we talk about the composition of agar, so uh, we use 1 to 2%. basically we can say 2% of the agar in the solid meat. Okay, if we are making or preparing a media, okay, so we mix. So about the concentration, we use 2% agar in the solid medium and 0.5 to 0, sorry, 0.2 to 0.5% in the case of semi-solid medium. Okay, and in liquid medium there is no agar. So we can directly say agar is a solidifying agent and made up of or from the seaweeds. Okay, and now preparation of agar media. So how we prepare a agar medium? Okay, basically how we prepare a culture medium, okay, or prepare a agar medium. Okay, so we just prepare a broth. Okay, broth means liquid medium is there. Okay, we prepare a broth like nutrient broth. Okay, and then we at the time of preparation we just add 2% of agar. So the broth means the liquid medium. So when we add 2% of agar, so it converts into the solid medium. Okay, so we can say it is a agar. Now like nutrient growth is a liquid medium, all the basic constituent is there, but there is no agar, so it is a broth, it is in the liquid form. But when we add the 2% of agar, so it converts into the solid phase. Now we can say it is a nutrient agar. Okay, now types of culture media, or we can say classification. Okay, so based on physical state, liquid, solid, and there is one more semi-solid. Okay, uh, we missed it, but there is first one is liquid, second one is solid, and there is semi-solid medium also there. Okay, and on the basis of presence of molecular oxygen and reducing substance in the media. So in on the basis of oxygen molecules, we divide the medium into two types, like aerobic and anaerobic. So bacteria, those uh, those bacteria are aerobic. Okay, so we use aerobic medium for the aerobic bacteria, anaerobic bacteria. Okay, so some bacteria anaerobic means they grow in the presence of carbon dioxide and absence of oxygen. So uh, for those bacteria we use anaerobic medium. Different types of media. So there is based on nutritional factor. Okay, everyone know what's the nutritional factor. Okay, basically some nutrients, particular nutrients require the bacteria for their growth or to promote their growth. Okay, so based on nutrition factor, there is simple media, complex media, synthetic media, and special media. So now what is the meaning of simple media? Okay, so in the case of simple media, it contains minimum ingredients that support the growth of non-fastidious bacteria. Okay, so here we use basic constituent. We all learn about the basic constituent. So in the case of simple media, we just use the basic constituent and they promote the growth of non-fastidious bacteria, not the fastidious bacteria. They just promote or used to grow the non-fastidious bacteria. Like example is there peptone water. Okay. So what is the composition of peptone water? 1% peptone, NaCl 0.5%, and water. There is nutrient growth 1%, mist attract. When we okay, we know one by one. Peptone water. Okay. So what is the composition of peptone water? Peptone 1%, NaCl 0.5%, and water. And we just add the 1% of meat extract in the peptone water. It converts into nutrient growth. And if we add 2% agar, it converts into nutrient agar. Okay. Uses testing the non-fastidiousness of bacteria. Okay, so these media we use to check the fastidious or non-fastidious bacteria, because uh, only non-fastidious bacteria can grow in the on these types of medium like nutrient, agar, peptone, water. Okay, and non-fastidious bacteria grow, but there is no growth in the case of fastidious bacteria. If there is a fastidious bacteria in the clinical sample, so there is no growth. Base for preparation of many media. Okay, so we use as a base of the medium to prepare so many other culture medias used for studying the bacterial growth curve. Okay, so we can use or can we can use to the studying about the bacterial growth curve used for biochemical test, colony morphology, pigment demonstration to check motility of bacteria. Complex media, all media other than simple media are complex media. Okay, uh, simple medium is basic constituent. Okay, so other than simple media, all media are come under the complex media. Okay, so complex media have added special nutrient required for the growth of the bacteria like TCBS, thiosulfate, citrate, bile salt, sucrose, agar. And what is the composition? Sucrose, sodium citrate, thiosulfate, NaCl. Okay, and what is the uses? Isolation of Vibrio. Okay, special media. Now, in special media, we go to the first we go to the enriched media. Okay, we subdivided special media. Okay, so in the special media, first one is the enriched media. Okay, so what is the enriched media? When basal media is added. Okay, basal means simple media. Okay, when basal media is added with some nutrients such as blood, serum, or egg, it is called enriched media. Okay, when we add egg, serum, or blood in the basal media, then it convert into the enriched media. Okay, like blood agar. Blood agar is enriched media as well as indicator media. Composition is nutrient agar. See blood 5 to 10%. It is used routinely. This is the picture of blood agar. Chocolate agar, it is enriched media. Okay, so we just heat it, the blood agar at 55 degrees centigrade for 2 hours, so it convert into the chocolate. Okay, and now it is a fastidious bacteria. It supports to support the growth of fastidious bacteria like Neisseria and Pneumophilis influenza. BHIV, brain heart infusion growth, composition calf brain and cow heart, peptone and dextrose, salts, disodium phosphate and glucose used for blood culture. Okay, and two types is there, monophagic and biphagic. In the case of blood culture bottle, these are the two types, monophagic and biphagic. So this is the image of blood culture bottle, monophagic and biphagic. Loeffler serum is added for and is the medium. Okay, so like Corynebacterium diphtheria. Okay, okay. So, Loeffler serum we used to grow the Corynebacterium diphtheria. It is a type of bacterium. Enrichment media, some substances are incorporated in the liquid medium which have a stimulating effect on the bacteria to the grow on inhibiting its competitor. So it is also a type of selective medium. Okay, but they are liquid. If we talk about the selective medium, so selective mediums becomes solid. But when we talk about the enrichment media, so enrichment media are the in the they becomes in the form of liquid medium as well as they are the selective medium. Okay, like cooked meat broth, RCM. Okay, so cooked meat broth sorry, uh, there is cooked meat broth. Okay, and cooked meat broth, RCM we can say, or CMB we can say. So composition, nutrient growth, pre-digested cooked meat of ox heart and uses of culture of anaerobic bacteria. Okay, so in the enrichment media, we can say RCM, cooked meat broth, and they are especially used to grow the anaerobic bacteria. Okay, so this is the picture of cooked meat broth. Now tetrathionate broth. nutrient growth, sodium thiosulfate, calcium carbonate, and iodine solution is there. So basically, they use to grow the typhoid and paratyphoid bacilli. Selenite egg growth. Okay, they also used to grow the salmonella. This is the picture of selenite egg growth. Alkaline peptone water used to grow the Vibrio cholera. Okay, composition peptone water and pH is 9.0. Okay, so Vibrio can survive in the high pH, but other bacteria cannot survive at the high pH. Okay, so we can use the peptone water with pH of 9.0 to isolate or to differentiate the Vibrio cholera. Gram-negative growth used for isolation of Shigella, strong inhibitory action against gram-positive bacteria because of its high content of citrate and deoxycholate. Selective media. Okay, so these media are used to isolate a particular bacterium from special specimen where mixed bacterial flora is expected. Okay, so selective media basically we can say when we take urine sample or stool sample and we just want to grow a particular bacteria. Okay, where then we use selective media. Okay, so selective media just promote a type of bacterial growth and inhibits the growth of other bacterium, okay, present in the clinical sample. Example of selective media DCA. It contains nutrient agar, sodium deoxycholate, sodium citrate, lactose nucleotide. Okay, so when we uh, culture the sample onto the DCAs, if there is salmonella and Shigella is there, okay, so growth will be there. If there is no salmonella and Shigella and other bacteria is there, so there will be no growth. Okay, or in the case of salmonella and Shigella and other bacterium is also there, so what happened, bacteria just suppress the growth of the other bacteria and just promote the growth of salmonella and Shigella. Algae medium, so just algae medium used for the mycobacterium tuberculosis. It is also a selective medium used to grow the MT, mycobacterium tuberculosis. BSA, wild salt agar. It is also selective media used for Vibrio cholera. This is a differential media. When a media contains substance which help to distinguish differing characteristics of bacteria, it is called differential media. If we talk about the McConkey's medium, there is so many media: blood agar, chocolate agar, McConkey agar, nutrient agar. Okay, so if we talk about the McConkey's medium, so McConkey's medium just differentiate the bacteria on the basis of it's give the color to the colony. If there is bacteria lactose fermenter, so it's lactose fermenter bacteria gives pink color colony, and in the case of non-lactose fermenter bacteria, so in the case of non-lactose fermenter bacteria, colony will be colorless onto the McConkey's agar. So it is a differential media, and also we can say it is a indicator media also. Why? Because it just indicate by the clearing of the colonies. Okay, Clade agar, cysteine lactose electrolyte deficient agar. Okay, it is also a differential media. It is used as an alternative to combination of blood agar and chocolate agar. It less inhibited than McConkey's agar, support GPV, gram positive bacterial growth, or advantage over blood agar. It can prevent swarming of Proteus. Okay, so Proteus is a bacteria that gives the swarming growth. Okay, on the blood agar. Swarming growth just growth is spread all over the medium. Okay, and we can say if there is more than one bacteria, so swarming growth of Proteus. Okay, they cover the colony of the other bacteria. Chilied agar, indicator medium. Okay, so in the case of basal biliary medium, okay, so in the case of Salmonella typhi, produce black metallic seal colony. Okay, if we put a sample or sample onto the Wilson Miller medium, and Salmi type is there. So it gives black metallic zinc or black metallic zinc colony. In the case of potassium tellurite agar, if bacteria is diphtheria, okay, so they also produce black colonies. In the case of Mekong agar indicate lactose fermenter properties, either bacteria lactose fermenter or non-lactose fermenter. Now, transport media. A transport medium is a holding medium designed to preserve the viability of microorganism in the specimen, but not allow multiplication. Okay, so they just preserve the bacteria or viability of an organism, but they cannot allow multiplication of a particular bacteria. Okay, so why we need transport medium? Okay, if we collect a sample in our college, okay, and there is no facility to test a uh, particular sample, so we send it to the another laboratory. So there is a time delay between the sample collection and the testing. So for the preservation of sample, for the preservation of viability, okay, we use transport medium. Stuart's transport medium is there, okay, used for gonococci, and buffered glycerol saline transport medium is there for use the enteric bacilli like E. coli, Klebsiella, E. coli or Salmonella वगैरह वगैरह, and carivaliar medium for Vibrio cholera, okay, and Bankerterman ramacrison used for the Vibrio cholera also. Now anaerobic medium. Uh, Anaerobic medium means like thioglycolate broth, okay, smoked meat broth, neomycin blood agar, anaerobic blood agar, and phenyl ethyl agar we use as the anaerobic to grow the anaerobic bacteria. Okay, they just produce a anaerobic medium to grow a anaerobic bacteria. Media for special use like MHA, Mirror Hilton agar used for AST or ABST by the disk diffusion method. Learn it into the we already learn it already into the culture methods. Okay, synthetic medium. Okay, so these medium are the purely made up of chemical with the exact known composition is there. Okay, we prepare a medium by the chemical and exact composition is known. Okay, there are used for studies such as metabolic requirement of a bacterium. Simple synthetic medium is there. They provide the basic essential for the growth of many non-fastidious heterotrophs. Do not support the growth of fastidious bacteria. Contains and they can basically simple synthetic media contains carbon, glucose, lactose, inorganic source of nitrogen in the form of ammonia, chloride, and phosphate. Now there is complex synthetic media. Addition to the simple synthetic media, certain amino acids, purines, pyrimidines, and growth factor. So simple synthetic media just convert into the complex synthetic media like simple media and complex media. We discussed about the medium. Duvose medium with two natives used to cultivate MTV species. Okay, there is a medium, Duvose medium with two natives. Okay, it is a example of synthetic medium. Okay, thank you.